You use muscles every day to do activities. You use muscles to breathe, circulate blood, and to move your arm to take notes. Your cardiac and smooth muscle tissues are involuntary, meaning you do not control your actions. Skeletal muscles work under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylinder-like cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of a signal change. This is where a synaptic bulb of an axon terminal and muscle fibers connect. Muscle fibers are composed of myofibrils. A myofibril contains sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thin or thick filaments changing in length. Skeletal muscle, its striated appearance. The muscle contracts when these filaments slide past each other. A thick filament is called a myosin, which is anchored at the center of the sarcomere called the M-line. A thick filament is composed of proteins called actin, which are anchored to the Z-line on the outer edge of the sarcomere. Because the actin filaments are anchored to the Z-line, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Muscle contractions are controlled by the action of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding site on actin. When calcium ions are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to the binding site on actin, forming a cross-bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. A contraction begins when a bond ATP is hydrolyzed to ADP and phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross-bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M-line, thereby shortening the sarcomere. ADP and the phosphate are released during the power stroke. The myosin remains attached to actin until new molecules of ATP binds, freeing the myosin to either go through another cycle of bonding and more contractions or remain unattached to allow the muscle to relax.
I need to watch when I'm submitting. She doesn't have time to regrade everybody's. I think it's only you. Okay.
Where should I put it?
I can cut stuff out, but. After the human body has been dead for 2 to 3 hours, rigor mortis occurs. Rigor mortis happens due to the overproduction of calcium and not enough ATP. So the calcium opens the binding sites for it to happen, but the ATP is not available to relax the binding sites for the next contraction. Therefore, the muscles stay in their quote-unquote locked positions, and this gives the stiff appearance in the body. In the sarcomere, the ATP is usually used to relax the myosin actin binding site to create the contraction and be able to start a new one. In rigor mortis, there is no ATP available to relax the myosin actin binding site, therefore, the cross bridge cannot detach from the actin. This results in the body and the muscles being in a locked position, giving off a stiff appearance.